However, before talking about the cellular responses of these systems, the general anatomy and physiology involved in this process will be described. The immune system involves cells cloning themselves, activating other cells, and warning still other cells about dangers in their environment. The first lines of defense are the skin and mucous membranes, but they seem to respond forcefully to body cells under stress, such as those that have been invaded by a virus. Once the target is chosen, the NK cells release chemicals that destroy the cell as well as any pathogen within it. Other warriors in the innate system are not fully developed cells, but rather protein chains. There are about 20 different proteins called complement proteins, which are produced by the liver. Normally, they remain inactive and patrol through the blood and pathways. A complement protein called C3 breaks up into C3a and C3b. C3b affixes itself to the surface of a pathogen, such as a bacterium, and then lures more C3b proteins to the microbe so it can be completely engulfed. A membrane attack complex, or MAC, occurs when a complement protein attaches to the membrane of the pathogen's cell and drives a hole right into that cell and kills it. Mannose-binding lectin, or MBL. Mannose is a carbohydrate molecule found on the surface of many viruses, bacteria, and yeasts. MBL binds with yet another protein, and together they attach to the mannose on the surface of the invader. At that time, the MBL and protein attract the C3 complement, which breaks into C3bs, and together they attack the membrane of the pathogen, then destroy its cell. The third pathway involves antibodies, which are proteins and two heavy chains that are joined together in a Y-like shape. At the ends of the two arms of the Y, proteins that are on the hands can grasp the proteins on the surface of the invader so the antibody and antigen can bind together. The bottom of the Y is known as the Fc region. Macrophages and other WBCs have receptors that can recognize this Fc region once the invaders are attached to the antibody. These killer cells are then activated so they can toll-like receptor, or TLR. TLRs are carried on the membranes of many leukocytes, such as dendritic cells and macrophages. There are many forms of TLRs which can recognize bacteria, yeasts, and even some viruses. When a leukocyte containing a TLR discovers a bacterium, the protein helps initiate a second major process of the counterattack, inflammation. The symptoms of inflammation, including erythema, swelling, heat, and pain, are all side effects of the war against the invaders, which has been initiated by the TLRs. Inflammation is a very complex process that includes increased blood flow, an influx of leukocytes to assault the bacterium. Some of their fragments spread into adjacent connective tissue or mucosa. When there, mast cells are activated. Mast cells, which contain granules from basophils, a type of white blood cell, are found in connective tissue and release histamine and other substances during inflammatory and allergic reactions. The antibodies mark bacteria or other invaders. Leukocytes, such as macrophages and deadly complement proteins, can find them for the kill. Because IgM antibodies appear fairly early in an infection, they are useful in the diagnosis of the particular invading organism. Also, IgM antibodies cannot cross over the placenta, so if they are present in a neonate's blood, designated as very simple microorganisms or extremely complex molecules. They have a protein coat surrounding an RNA or DNA core of genetic material, but no semi-permeable membrane. They are only capable of growth and multiplying when in living cells. Because of these anatomical and physiological features, once attached to the host cell, the virus injects its nucleic acid into it. This acid then takes control over the normal function of that cell and produces multiple copies of the virus's protein coat and nucleic acid, which are then assembled into new viruses. As the host cell becomes crowded with these new clones, it bursts and dies, sending the viruses in search of a new host. This process is called Fas Ligands, which is on the surface of the killer T cell. This molecule can find its complementary molecule on the surface of an infected cell and fit into it like a key into a lock. When attached to the body cell, Fas Ligands triggers the cell to self-destruct. If this battle is unsuccessful, the adaptive immune system joins the fight. B cells, specific to the invading virus, mature and start marking the pathogens with antibodies. This process works well against viruses that are floating within the blood. However, some viruses spend their time hidden within the cells of the body. Before these viruses can be destroyed, they release new copies of themselves through the cell wall. These clones burrow into other cells before warriors can attack them. The program will look at autoimmune diseases, or pathologies in which the body literally attacks itself.